I thought we should talk genetics. I think it's time we got a bit of an education, maybe, because you get taken for a ride by these so-called we found the missing link and all this sort of stuff. You get taken for a ride by these scientists who really they're just trying to make a name for themselves. Okay. Genetics. Okay, so you've got your body has got cells in it. Okay. Heaps of them. The numbers are pretty staggering. Just in 1 second alone, a trillion blood cells are being formed by your bone marrow. Okay, so a trillion a second. So that's just your cells from your blood, from your bone marrow. So the numbers utterly staggering. You are fearfully and wonderfully made. It's an incredible thing. The cells all have they're like a giant spaceship, really, with portals on them. And the portals can open and close and allow information, materials in, materials out. Seems to be an intelligence behind, or maybe not intelligence, but it's controlled. Okay, there's a control behind why let this in, why not let this in. All these mechanisms. It's a lot of stuff to make. A lot of proteins needed just to form that incredible thing. Inside you have areas with different parts, and they're all very interesting. But the one I want to go to is the nucleus. In the nucleus is where we find the DNA. Called the they call it heaps of names. They call it the double helix and all this sort of thing. It basically looks like a ladder with ruts going across, like you'd walk up a ladder. But it's twisted. The ladder is twisted around. It's not a perfect gaps between the twists. There's actually tight twists and then slightly open and tight, and they've all got names. So the helix itself, the base on the side, called the backbone, it's made of glucose and sugar and phosphate. So it's a phosphate backbone, and across the middle, the rings, they're called the base pairs. They're made up of different neutrinous, no, very tired. Oh, I'm gonna have a hard time. I'll come back to that one. Anyway, they're called nucleotides. Okay, and they have different types. And the nitrinous bases on the end. Okay, they have 4 types. There's the I think it's adenine, cytosine. What is the other one? Tanamine, and guamine. And they pair up. Basically, 2 runs, and they pair up, and there's these hydrogen links between them. Okay, bonds that form, which break apart like a zip when they copy each other. So they pair up. A goes to T, adenine goes to tanine. Oh man, good grief. Cytosine goes to guamine. I'm really tired. Anyway, they match up, and they are called the base pairs that are going across. It's very much like a code. Every 3 is every 3 of them, when it's transcribed, which means it's read, this little how do you do it? Okay, just think there's like this thing that attaches to it, and it starts reading, it runs along, zips along, and it starts reading reading off the line, the run line. Okay, it unzips it and reads it off, and then as it's running along, zipping off, it's creating a copy of one side called RNA. Well, it's actually mRNA, messenger RNA. It comes off. It's not quite exactly the same. It's a slightly different substance, and it uses U instead of T. Okay, so the letters are slightly different. The RNA, what comes off the copy from the DNA. So anyway, the nitrous. Anyway, it's got lots of nitrogen areas, and okay, adenine and all that. I can't say nitrogenous base. Anyway, now you've got all that, and the different chemicals come along and they attach, and they've got these regulator things. How much of this, how much of these stuffs are we going to make, and what are we going to do? How far are we going to transcribe, which is like copy of this DNA strand? And so it's all being regulated by something. And it used to be called junk DNA. They found out, oh no, this has actually got a use. But they've also found these things called epigenetics. And epigenetics is like outside chemicals and things come along and attach themselves on. And it can be from your, it's a big area. Your environment seems to actually affect what's going to come along and attach when it starts reading it off and zipping off, and it's telling it where to start, where to stop, based on your environment as well, not just the DNA. So sometimes your environment chemicals can actually control how much or how little or of what's going to be copied, and that's a very interesting epigenetics. Okay, DNA's got a code in it. It has a code which is incredible. Now I know a bit about coding, and it takes an intelligent person to do a computer programming code. Zeros and ones, lots of intelligence needed to write a program that does something. Now, these genetic base pairs, every 3 is a codon. It can be transcribed, which is the copying part, and then what happens is the RNA is gets taken out of the nucleus, goes into it's not ectoplasm, I always say that, it's something else. It's not ectoplasm. Anyway, it's some plasm. I can't remember. Anyway, it goes into the plasm part, and then a ribosome, which is like a little mini factory, comes along, and lots of proteins, other proteins come along, and you've got these tRNA, which is like transport RNA little guys, and they come along with amino acids. Okay, and they come along to the ribosome, and what's happening is the RNA comes out, squiggles along, the ribosome locks on, finds the place where it's going to read it from. It's like got these starting little codes, okay, and it starts reading and transcribing along that RNA. The amino acids come into the ribosome, they match up like a key looking for which one it fits. When it fits, it lets go of the amino acid, and then the next one comes along, and then the next one, and then basically the first one takes off, and it's just like this little chain reaction, and they're all fitting, going, oh yeah, you're right, and then making the amino acid chain. Okay, so it's fitting, saying, oh, this is the key, this amino acid goes here, then the next one, and it joins to the amino acid chain. So it's doing that. It's like a very interesting little production thing. Now they thought that DNA was only useful for making proteins, okay, and that proteins only came from 3 genes, 3 base pairs makes matches to an amino acid. And that's what they thought. So they looked through the DNA and going, oh yeah, there's the ones that match the amino acid. There's 20 amino acids to choose from, and the combinations of those amino acids put together is where we get the proteins, and they start folding up into these useful little proteins and molecules that run around and do stuff in the cell. Okay, So it's all very amazing. I love it. I think it's incredible business stuff. I don't know if you remember the name of that plasm. Anyway, then we okay. So it all copies, and it's called, and the part of zipping down the DNA is called fancy word called transcribing. Okay, big fancy thing. And then the RNA coming out or going to the ribosome and getting read is the translating part, and it's getting translated into a protein. Now, scientists thought they had it down pat, didn't they? But just in the last number of years, 5 to 10 years, not even that actually, they have found, since 2005 actually, 2003 onwards to 2005, they really started making headway, all the way to 2012. They made some big discoveries. Now, what I just told you was the old way they used to think about genetics, okay? It was like beads on a string, okay? That method. No, it's not actually how they've discovered it works now, okay? So all them years and years of theories are pretty outdated. But what I told you wasn't actually wrong or anything, it was just not quite accurate. So what happens, they were thinking there's this thing called a 3' and a 5' on the phosphate backbones, okay? And they're anti-sense. They go in opposite directions because of the phosphate at one end. So when you're adding something on, like a nucleotide on, you can only add it on one end. That's all. Okay, so it's just the nucleotides, which are the base pairs, you know, adenine or methionine, cytosine, guanine. Anyway, it only goes on one end. They thought it only read in one direction when it transcribed. It only peeled off, it only read part of the DNA on one side. What they found is it actually transcribes both sides in the DNA, goes down both ones in both directions. It's a new one. It is not just reading 3 for making a protein, but sometimes it's reading the 3 base pairs in reverse order, so forwards and backwards, and using them, and they're making different proteins by reading it forward and backwards. They're also finding it's jumping genes, so it's maybe going this gene and then jumping 5, grabbing that one, and then grabbing that one, and it's actually having these amazing patterns, and then it's transcribing them, and they're making genes, and it's grabbing sometimes bits of genes here, going through, because the genome's got like 25,000 or something genes they found, so 22, sorry, goes to another chromosome area, part of the genome, grabs a bit of genes, okay, and it starts reading that, and it combines it with that part from over there, puts it together, makes a bit of another proteins from combinations throughout the whole thing. It's just incredibly insane, and they're like, how is this possible? This is way far more complicated code. When the scientists mapped and said, we've mapped, was it 2003 that we've mapped the genome? Oh, we've done it, you know. They've only actually mapped the skeleton. Okay, the code of DNA is so complicated, it's mind-boggling for them. It's just way more complicated than they ever imagined. Yeah, so it's thrown DNA, genetics into big turmoil. So if you haven't learned genetics before, that stuff I was just talking about, what is the genome? You might not know. What is mapping the genome? Why are you saying genes and genome are the same? No. The genome is the entire thing, okay? All the genes. And it's split up in little areas, as I was saying, that make proteins, and they're all identified. They are, the genes are little parts, and just think of the genome as the whole shaboodle. And you know when they say, oh yeah, we're like chimpanzees and all that, chimpanzees' genomes are like 13% larger, okay? And they're finding it not as similar as they were thinking in the past, okay? So it's all very interesting. Epigenetics, environment controlling, what's going to be transcribed, how much is going to be transcribed. And our environment actually is affecting our DNA, protein development, cell structure and building in our body. We are far more complicated than they ever imagined. What does this mean for evolution? Well, it's actually a real big problem because it's meaning um, the code is far more complicated. Now, like in information science, for instance, if you're sending a computer message to another computer, you can send it in, because computers are digital, they've got on and off switches. The binary, that's how they're stored in transistors, current running through them, so an on and off state, just like a light switch. Okay, so you've got millions and millions of them in your computer, and you send a message. You can send it as in a pulse, on or off signal. You can do it using voltage and electricity. You can do it with light, on and off signals, with lasers or LEDs. You can do it with sound. Okay, so any on or off signal, yes, the sound's on, yes, it's off, or whatever, any change in current or anything can be used to send a binary pattern. Binary is just the number system we use because it's easy, because it's only two states in digital devices, are an on or off state, or so binary has two numbers, 0 and 1. So we use that for to describe, to show what digital devices are, storing stuff. So anyway, you're sending the information, the message, along a transmission media. Now the transmission media doesn't actually make any. It really doesn't make any difference what you're sending along. I could send it along a cable, Cat5 cable, copper twisted pair, which is what most land networks, like area networks, are made out of, pretty cheap stuff. Or I could send it way across the country using fiber optics, using the lasers, just the same 0 and 1 on-off states of whatever it is. I could send it by satellite. I could send it by X-ray. I could send it by radio signals. I could send it by does it really matter? Because the message is actually still there. The transmission medium can change. It doesn't really matter how it's getting across. And that's what the basis of SETI is, search for extraterrestrial life. They're going, we're listening for a signal that shows intelligence. Now, DNA has like binary code, and it has instruction code for making stuff. It has actually information in a transmission media, which is the DNA helix. Does it matter what it's stored in? The medium? No. Information is information. Now, where on planet Earth do we find information? Information has only been, by scientific observation, and we only know one way information can exist, and that has been by intelligence. Search for extraterrestrial signals. They're listening for a pattern that shows intelligence. Okay, and they're not worried about the transmission media. They want a pattern that shows intelligence. But in all of us is a DNA, which is this information. Okay. Now, the whole thing about genetics, and you say, well, how do we evolve? Well, natural selection. It all could be possible. God could have used natural selection. That's mutations in the DNA. Now, mutations in the DNA. There's different types of mutations. There's point mutation. There's all different ones. Okay. Now they're like changing the letters and stuff with the amino acids when they're making them. Some of them have no effect. Some of them shift the message along, completely destroy the proteins, and then your cell basically might not, the protein might not do what it's supposed to do, and you get big trouble. Some of them, add, there's insertion, deletion, or all these different ones called point mutations. And so you've got these issues, and what could happen is mutations, though. Let's just look at it for a second. If I'm sending a message, a radio signal, across the airways, so there's my transmission, there's my information being sent, and then we put noise or interference up, so it interferes with the signal. That's like mutations. Does that add any information to the signal. No, it only corrupts the signal. So mutations haven't been known to actually add any information to DNA. That's the problem. The mechanism that has been proposed for evolution to work by natural selection is mutations. Now, mutations don't add information; they only corrupt information. Okay, and that is a big problem. That means you're going to have radio signals that are getting garbled by junk, and they're never ever going to improve by putting more junk and more of junk, like interference, more interference, more interference. It's going to ruin the signal more and more. What do we observe then? What do we actually observe? We observe more and more interference or mutations in the DNA, and have any of them had any benefits? Now, if you ask a biologist, can you name an actual mutation that's actually a beneficial mutation? Because, you know, this is your mechanism for evolution apparently to have happened by natural selection. So how? Is there a mutation you've actually observed that's improving? And they'll come out with a couple ones they might throw at you. They'll do sickle cell and anemia. Now, people die from that, okay? It does give you a cure to malaria, possibly, but you actually still get really, really sick and you're probably going to die. Does it make you survival of the fittest? Does it make you stronger? I argue, oh, you don't get malaria, but if you're going to die, it's from the sickle cell anemia, is it actually beneficial mutation? It's a very arguable one. But hey, if that is the best they can do, okay, out of all the mutations, if that's the best they can do, then are we evolving upwards by mutations? You know, can it happen with all the amazing stuff that we've got? Interesting. Okay, so yeah, beneficial mutations a bit of an issue there. So that, yeah, and so creationists have jumped on and they've gone, well, yeah, mutations don't add information anyway. Even if they were beneficial, they're still not adding any information to the original information. So how can you have a single cell organism that is using natural selection to select from its DNA what it exists to make choices? It can only select from what information is already there in the DNA. It can't. It's got no other information to select from. Okay, so it can only select from. It's not adding. Natural selection is not adding information to select from. So is it a mechanism using mutations? No, it fails. So it's been a real problem. I mean, it actually should be, hey, we're done and dusted. Yeah, you guys lost. Thanks for playing. But no, they're good at fighting back. So molecular biology. I'm having trouble. You know, biology came up with, hey, let's put in instead the idea that there's these type of viruses. Now I think they're called retro viruses. Anyway, they've got some fancy name, retro blah 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 viruses. Anyway, they get into the DNA and they insert themselves in there and they're a virus and so they're saying, hey, we've got some adding information now by this virus inserting itself in your DNA. Now there is a lot of things you could go down here. You could say, well, does that mean God made slots for them originally when he designed? Is that what it means? You know, like there's avenues to explore. Okay, so evolutionists have been trying to pull themselves out of the mire and say, yeah, we found it. We found some way of getting some information using the retro viruses. And yeah, I'm interested because a lot of creationists are looking at that, and they are actually starting to look at that very, very seriously now. And already the threads are starting to show, starting to come a part of it. I'm still watching the space, Because, as you know, every single thing that's been proposed in evolution has actually gone down big time. So I don't get all upset just because evolution tries to get some come up with some scheme to hold on to their belief system. They're very resilient, you could say. Okay, but it's interesting. Genetics is a fascinating topic. Okay, hope you learned a little bit, and maybe you can keep up when you hear these evolutionary debates. You can actually understand what they're going on about when they talk about genetics. You can go, oh yeah, whatever. And if they mention junk DNA, just laugh at them because they are so far behind in keeping up with current breakthroughs in science. Okay, and vestigial organs. I heard a science teacher actually say that, and I could not believe it. Okay, shoot, that one was well and truly buried a while ago. But yeah, I think Richard Dawkins goes. One of his ones recently has been that poor design. You know, the eye. If it was made, the optic nerve should be behind, not in front. Goes in, it's blocking part of the vision. It's way better behind. But he has no understanding at all of how the eye should work. And people who have actually got actual far more qualifications in that area than him have pointed out he is dead wrong. The design is far more efficient than if he had tried to design it that way. So his whole thing about, oh yeah, poorly designed, is just idiotic because he doesn't. Yeah, they didn't do their homework on that one.